Hey guys, so I hope everybody is doing all right. I'm really sorry that I couldn't see you guys for the rest of this week, and I know it's going to be a block day. Hopefully you've watched my instructions video before you're starting to watch this one. It'll tell you exactly what you're going to do for the whole rest of the day. So we ended off on Tuesday with talking about our different substances. And I think I was a little bit rushed on Friday, so I want to or Tuesday, sorry, you know me, days of the week. And I want to take a step back and kind of go over this again, just really briefly. So there are 4 major macromolecules that are in biology that we really use a lot. You need to consume these all in some way, and they are all in food products of some sort. And the reason that you need to do this is that everything else in your whole body is really built around these 4 things. So before we talk about what they are, I want to talk about the difference between a polymer and a monomer. So a monomer is something that there's just one of. So if you have a monocle, that's going to be one glass. So instead of having both of your glasses, or both of the lenses in your glasses, it would just be one. So that's what mono means. Mono means one. Poly, on the other hand, means many. So you've got a bunch of different things all stuck together. So every single compound that we're talking about today has monomers and polymers. So a polymer of a carbohydrate is just a bunch of monomers of the carbohydrate stuck together. Imagine the difference between a single link of chain and a whole big string. So each one of those links is a monomer, the whole chain is a polymer. So it's kind of the difference between monomer and polymer. So for carbohydrates, our monomers are going to be called monosaccharides, and you really do, guys, need to know what the monomers and polymers of each of these compounds are. It's really important to talk about. So the monosaccharides, the 2 biggies, are glucose and fructose. Our polymers are going to be called polysaccharides. So as you can kind of see, there's a little bit of a naming convention here. Mono for one, poly for multiples, and then saccharide is just refers to sugar or carbohydrates. So our main ones that we know here are sucrose, starch, not stark, we're not in Game of Thrones, starch, and glycogen. Sucrose, that's going to be your table sugar. So lipids. Lipids are otherwise known as fats. We talk about things like oils, bacon grease, things like that. And there's a bunch of other different types of lipids, which we'll also talk about, but that's generally the first 2 that people think of is fats and oils. So we get away from the kind of nomenclature here, or the traditional namings like we had with monosaccharide and polysaccharide. There's not quite as much a monomer for lipids. We say that they're glycerol and fatty acids, but that's only sort of true, and you'll see why in just a minute. And then we've also got saturated versus unsaturated fats, which is kind of true and kind of not, which you'll see in a minute. So proteins. This is the one that people are most familiar with, I've found, because we call meat protein. We say that protein is an important part of your diet. All those things we already talked about, so people know a little bit more about them. The monomer of a protein, and you absolutely, if you don't know any of the others, please know the monomers of proteins and nucleic acids. It will be coming up just tons. So the monomer of a protein is an amino acid. When you put a bunch of amino acids together, you get a polypeptide. Whenever you see pept, that's going to be referring to protein. So polypeptide, or we'll sometimes just call it a protein because we get lazy like that sometimes. And then the last one is nucleic acids. Your nucleic acid is DNA. That's going to be the main one that we talked about this year. DNA stands for deoxyribonucleic acid, and if you're in one of my classes, we've already practiced saying that. If you're not, we will be saying that next unit. The monomer of a nucleic acid is a nucleotide. Nucleotides are made of 3 different things. You don't need to know that just yet. We will get there. So, moving right along. Carbohydrates, the basics. So these are going to be used for short-term energy in the body. Think blood sugar. When you're not feeling so great, your blood sugar is low, you're tired, you're kind of antsy, things like that. But we also use them for structure in the body as well. So these can be consumed in the form of sweets, breads, pastas, vegetables, fruits, potatoes have a lot of carbohydrates in them, things like that. The Atkins diet is a diet that takes you off of carbohydrates and forces you to eat things other than carbs. So anything that you couldn't eat on the Atkins diet, it's going to be a carbohydrate. And then we already talked about the monomer and polymer. So this is a good picture to talk about monomers and polymers with. So we have glucose. You can see that glucose is one solid ring, so it's just one of them. When we get into maltose, so this is the monomer. When we get into maltose, this is a polymer. This is a special type of polymer called a dimer because there's only two of them and di means two. So this is technically a disaccharide, but again, slightly more information than you need. So this is one molecule of glucose and this is one molecule of glucose. When you connect them together, it goes from being a monomer or a monosaccharide to a polymer or a polysaccharide. Again, in this case, it's disaccharide. So, carbohydrates, the monosaccharides. So, carbohydrate is a compound that contains carbon, hydrogen, and oxygen. The ratio of hydrogen and oxygen is in the same water. So, there's always going to be 2 hydrogens to every 1 oxygen. And I'm going to explain this better when I'm actually with you guys. Hopefully somebody will remember to ask me this, or I'll write myself a note. Whenever you're writing it out, there is a 1 to 2 to 1 ratio. So the formula for glucose is C6H12O6. So for every 1 carbon, there's 2 hydrogens and 1 oxygen. So these are our big 3: glucose, fructose, which is going to be what's found in fruits, and galactose. Our disaccharides, they form a covalent bond between them. A covalent bond is simply a bond that shares electrons. And next unit, we're going to be going into the bond types. There's 3 types of disaccharides. When we talked about disaccharides being 2, sucrose is glucose and fructose. This is what your table sugar generally is. So polysaccharides. I talked about how we use short bursts are going to be more your mono and disaccharides. Polysaccharides like this one, this is glycogen, can be used for more long-term storage. This is how your body stores energy. So you actually use glycogen. And you can see this is a true polysaccharide. It's not a disaccharide, it's a true polysaccharide because there's a bunch of different rings all linked together. It looks like a fence, or it looks like a chain all linked together. So the most important structural polysaccharide is cellulose. Cellulose is the cell wall. You should kind of know that briefly from your other life science class. So anything that's going to be made out of a plant fiber, that's going to be made of cellulose. We cannot digest cellulose, so that's kind of why, that's what fiber comes from. Fiber is, wow, you guys just thought you were going to go to avoid my hiccup. Guess not. So fiber is plant cell walls. It's the cellulose that we cannot digest. Another one is found in the shells, the exoskeletons of crabs and lobsters. It's called chitin. So lipids. Lipids are our next one. So this is used for long-term energy storage. Gram for gram, lipids have twice the amount of energy that carbohydrates do. So 2 times the energy in a single gram. So that's why our bodies prefer to store energy in lipids. So it's also a large part of the cell membrane structure. The most important lipids are phospholipids and cholesterol. So you can see cholesterol over here. It looks almost like chicken wire all together. All cholesterol is going to look basically like this. We can consume lipids through fats and oils. This is kind of what fatty acid looks like. So phospholipids. I would draw this. The easiest way to draw this is to do one small circle here, another small circle, and then 2 lines off of it. It's the easiest way to draw it. And I want you to make sure that you label this as a phosphate. We'll talk about what phosphates are when I get back. And that these are 2 fatty acid tails. So phospholipids are similar to triglycerides, which we kind of skimmed over because you don't really need to know them. The phosphate end of the molecule likes water. So this right up here, it likes water. It's what we call hydrophilic. And the fatty acid down here does not like water. It does not want anything to do with it. It's a little bit bipolar of a molecule. So we've got this that loves water, this that hates water. So it has to arrange itself in a certain way in order to make everybody happy. And the way that it does that is it bands up with other phospholipids to form what's called a phospholipid bilayer. So you can see all of the hydrophilic heads are on the outside of this bilayer where there's water. And all of the insides are protected because they're so closely packed together. So the tails are on the inside, the heads are on the outside. So this is the structure of the cell membrane. This same structure goes all the way around the cell, and that's kind of what it looks like. And this is kind of where cholesterol comes into play as well. So if you just had a whole bunch of phospholipids, it would be very loosey-goosey. It'd be kind of moving all over the place. But cholesterol is an unusual type of lipid. Everything you've seen so far have been very linear. Cholesterol is a bunch of rings. And it actually is inside that plasma membrane. And I'm going to go back for just a second. So you take this molecule here and you stick it in between the phospholipids. So you stick it in there and it kind of helps to stabilize it. That way your cells aren't literally melting. And we can talk about that when I get back. It's also used to manufacture a class of hormones called steroids. No, not the anabolic, make Hulk mad kind of steroid, but the kind like testosterone and estrogen, progesterone, things like that. Now, there are some problems with having high blood pressure. With having high cholesterol, and that is high blood pressure and heart disease. Because that molecule is so kind of weirdly shaped, it can block your arteries and blood vessels. Not what you want. Alright, so proteins. This is the only slide on proteins because I actually have a whole nother video that I want you guys to watch and go through about proteins. So proteins are really large, really complex. Make sure you know that the monomer is a is an amino acid. Sorry, please don't write that down. The monomer of a protein is an amino acid, and the polymer is a polypeptide. These are held together by peptide bonds. Again, if you hear peptide anywhere in there, we're talking about proteins. So an enzyme is a type of protein that we will talk about later. In fact, we're going to talk about that next. So that will be page 16 for y'all. So we'll come back to that in just a sec. All right, nucleic acids, DNA and RNA. This is our entire next unit, so I'm not going to spend too much time on this. Just know that DNA and RNA are both types of nucleic acids. DNA is always found in the nucleus of the cell, and it's a double-stranded molecule. And again, we're going to go over this in excruciating detail, so if you're a little bit confused, you can send me a message on Schoology, but we're going to go over it. And then ribonucleic acid, RNA, is a single-stranded molecule, and it's found in a couple of different places. It works with DNA really closely. There are a few key differences, though, which we'll talk about. So this is what a nucleotide looks like. It is a sugar. And this can be any sugar. You can see this one is deoxyribose, but that's only for DNA. For RNA, it's a different one. A nitrogen base over here. There's 4 of them. We'll talk about them. And then a phosphate. So this is what a nucleotide looks like. It's made of 3 different pieces: a sugar, a nitrogenous base, and a phosphate. Please know those 3 things. You need to know that a nucleic acid is made of a sugar, a nitrogenous or a nitrogen base, and a phosphate. And then you can see ribonucleic acid here. DNA can only be found in the nucleus, whereas RNA can be found in the cytoplasm of the cell. And I'm sure these words are all Greek to you. Don't worry about it. We're going to cover it. I've only got about a minute left in this film. It cuts me off after 15 minutes, so I'm kind of trying to wrap it up. But again, only difference between RNA and DNA in this instance is this sugar here. You can see that we've changed it for ribose instead of deoxyribose. So that's what's happening with that. At this point, you should have all of these notes written down on page 15 in your notebook. The next thing I would like you to do, turn the page, go to page 16, start by labeling it enzymes in there, and I will see you in the next video. Should be right below this one in Schoology.